Imagine you have a patient, right? Someone with severe treatment-resistant depression. You know the scenario. You've taken their history, you've spent months building a therapeutic alliance, and over the past year, you've tried, 3 different SSRIs?
Easily 3. Yeah, and you probably augmented with typical agents.
Right, exactly. And nothing is working. The patient sits across from you in the consultation room feeling like a profound failure. They are utterly convinced their mind is permanently broken.
It's a heartbreaking scenario. But, what if the solution to their psychiatric distress isn't actually another traditional psychotropic drug? What if instead we need to be looking at a protein produced by their liver?
Which just sounds wild, doesn't it? But that is the mystery we are unraveling today. So, welcome to this deep dive. For the psychiatry trainees, the registrars, and the clinicians listening right now, our anchor premise today is foundational.
What?
Psychiatry is the study of how the brain, the mind, and experience interact, and crucially, how that profoundly intricate process can go wrong.
And today, we are pulling a very specific thread. We're dismantling that archaic notion of the blood-brain barrier as this impenetrable moat.
The classic VIP room metaphor.
Yes, exactly. The VIP room in the nightclub.
Which we just need to throw in the bin. I mean, in medical school, the blood-brain barrier is often taught as this absolute fortress wall. The brain is this pristine monarch locked away in an ivory tower, completely untouched by the muddy, chaotic mess of the body's systemic illnesses.
We love to compartmentalize, don't we? Physical etiology over here, psychiatric etiology over there, neat, separate wards.
It's intellectually comforting.
It is comforting, but it's biologically false. I mean, the modern understanding of neuroimmunology paints a completely different picture. The fortress is actually incredibly porous.
Right.
It's in a constant dynamic and sometimes frantic dialogue with the body's roving security forces, the immune system. And we aren't just talking about pathology here. We're talking about the normal regulation of your mood, your behavior, and your cognition.
Wait, so the brain isn't a VIP in a soundproof room at all. It's more like a control center constantly receiving frantic security briefings from the rest of the body.
Spot on. That is the exact reality.
And that is our structural spine today. We are drawing upon the brilliant concepts detailed in Chapter 143 of the comprehensive textbook of psychiatry, focusing on immune-brain interactions.
We're going to trace the breadcrumbs, essentially, from cellular mechanisms right up to clinical manifestations.
And finally, to how you, as a clinician, must shift your clinical reasoning. Because if the brain is constantly receiving these security briefings from the gut, the fat cells, the liver, we can't just treat the brain in isolation.
We absolutely can't. We have to understand the language of those security briefings. And to do that, we need to just briefly define the 2 main branches of the body's defense forces.
Right, the innate immune system and the adaptive or acquired immune system.
Exactly. And they operate on entirely different timelines and principles.
I always sort of conceptualize the innate system as the frontline paramedics and the beat cops. They are fast, they are nonspecific, and they don't really need a detailed dossier on a suspect to know that a riot is breaking out, right?
That captures their role perfectly.
Yeah.
The innate system provides that rapid initial response. We are looking at cells like macrophages, neutrophils, natural killer cells.
The patrollers.
Right. They maintain this constant background level of surveillance. But they aren't looking for, say, a specific strain of influenza. They are looking for broad patterns of destruction or foreign presence. They do this using pattern recognition receptors.
And the toll-like receptors are the most famous of that bunch, aren't they?
They are, yeah.
But what fascinates me is the evolutionary elegance of what these receptors are actually scanning for. They scan for Pampommy's pathogen-associated molecular patterns, like the lipopolysaccharides on a bacterial cell wall.
Yes.
But the other target is where psychiatry intersects with immunology, and that is DAMPs, danger-associated molecular patterns.
And this right here is the pivot point for our entire discussion today. DAMPs are not foreign invaders. DAMPs are the internal debris of our own cells.
Oh, wow.
Yeah. When a cell in your body undergoes severe stress or it dies a necrotic death rather than a quiet, programmed apoptotic one, it literally bursts open. It spills its internal contents into the extracellular space.
So we are talking about intracellular molecules that have suddenly found themselves outside the cell where they don't belong. Things like ATP, the energy currency, or heat shock proteins.
Or even fragments of mitochondrial DNA.
Wait, really? Mitochondrial DNA triggers an immune response?
Absolutely. Think about it evolutionarily. Mitochondria are essentially ancient bacteria that we incorporated into our cells eons ago.
Oh, right. So they look like bacteria to the immune system.
Exactly. So when mitochondrial DNA leaks out, The innate immune system perceives it as a bacterial invasion. When a patrolling macrophage encounters this leaked ATP or DNA, its toll-like receptors bind to it. This activates an intracellular protein complex called the inflammasome.
Specifically the NLRP3 inflammasome, right?
Yes, the NLRP3.
The emergency
A
siren.
very, very loud emergency siren. The inflammasome triggers a massive cascade of inflammation. But notice the crucial detail here. There is no infection.
None. There is no actual bacteria or virus.
None at all. It is entirely sterile inflammation. And this bridges the gap between psychological trauma and physical pathology.
Okay, let me pause on that because the clinical implication there is staggering. To the innate immune system, a severe psychological stressor, say, chronic childhood abuse or unrelenting financial panic, looks biochemically identical to a raging bacterial infection.
It does. The beat cops don't care about the source of the debris. They just see the debris from stressed neurons and endocrine cells, and they start throwing molecular tear gas.
That is incredible.
And the consequences linger far longer than we previously believed. I mean, the old dogma dictated that innate immunity had no memory whatsoever.
Right. It just reacted, cleared the debris, and reset to zero.
Exactly. We used to think only the adaptive T and B cells had memory. We now know that is demonstrably false.
You are referring to trained innate immunity. I know this is a relatively recent paradigm shift in the literature.
It is a massive shift. When innate immune cells are exposed to a severe stressor or a damp, they undergo epigenetic reprogramming. The histone packaging around their DNA is modified, which actually changes how their genes are transcribed for months afterwards.
So the macrophage holds a grudge.
It holds a serious grudge. It becomes hyperreactive. If it encounters a minor stressor 3 months later, it mounts a massive, exaggerated inflammatory response because its epigenetic baseline has been shifted.
It's akin to short-term memory, whereas the adaptive system provides the lifelong memory.
Exactly. Let's bring in the adaptive system because the interplay is vital. If the innate cells are the beat cops, the adaptive cells, the T and B lymphocytes, well, they are the highly specialized tactical units. They represent the crowning achievement of immune evolution.
Their specificity is mind-boggling. The receptors on a single clonal line of T cells are designed to recognize one very specific antigen out of literally billions of possibilities.
They are slow to deploy.
Right. Why are they so slow?
They are slow because they require a highly controlled verification process. Detecting an antigen is not sufficient to launch an attack. They require accessory signals co-stimulation.
Like a secondary verification.
Yes, usually provided by antigen-presenting cells like our innate macrophages.
It's the biological equivalent of a nuclear launch protocol. You need the antigen key and you need the accessory signal key from a commanding officer.
Exactly. It prevents the tactical units from accidentally launching a massive strike against the body's own healthy tissue.
Which would be an autoimmune disaster.
Precisely. When both keys are turned, the lymphocytes undergo clonal expansion. They multiply rapidly. B cells differentiate into plasma cells to churn out highly specific antibodies. Cytotoxic T cells hunt down infected cells.
But the real magic is what happens after the threat is gone, right? The memory cells.
The memory cells are brilliant. After the threat is neutralized, a subset of these specialized cells retreats into the reticuloendothelial organs, the spleen, the lymph nodes.
The vast reference libraries of the immune system.
They reside there, sometimes for the entire lifespan of the host. If that specific antigen ever breaches the perimeter again, the secondary response is instantaneous and overwhelming.
Okay, so we have the fast, blunt, innate system looking for cellular stress, and the slow, precise, adaptive system building lifelong tactical weapons. But let's unpack this. The brain is sitting all the way up in the skull. How do these security forces communicate their alarm to the central command?
That's the big question.
Right. If a macrophage in my gut detects a damp, how does it relay that systemic panic up to the brain?
Through the immune system's proprietary communication network, cytokines. These are large molecular weight proteins released by activated immune cells. You can view them as long-distance text messages.
And in the context of psychiatry, we are laser-focused on the pro-inflammatory cytokines, aren't we? Tumor necrosis factor, or TNF, interleukin-1, and interleukin-6.
TNF and IL-1 are the initial alarm bells. They are the principal mediators responding to immediate threats, and they act as endogenous pyrogens.
They cause fire, they initiate the fever response.
They travel to the hypothalamus, stimulate the synthesis of prostaglandins, and force the brain to crank up the body's core thermostat. They also induce cachexia.
That's the profound muscle wasting and appetite suppression we see in severe chronic illness, right?
Yes. But the systemic cascade really gains momentum when the signal is passed to interleukin-6.
Because IL-6 is the courier that travels directly to the liver.
The liver acts as the central manufacturing hub for acute phase proteins. When IL-6 binds to the hepatocytes in the liver, it essentially forces them to halt their normal operations. They have to begin churning out proteins designed to isolate pathogens and limit tissue damage.
And the most clinically relevant of these proteins for us, without a doubt, is C-reactive protein, CRP.
Absolutely.
For the clinician listening, this is where the biochemistry translates directly into your daily practice. Why do we care about a protein synthesized in the liver when we are trying to treat a mood disorder?
Because measuring cytokines directly in human plasma is notoriously difficult. I mean, they have short half-lives, they fluctuate wildly by the minute, and standard hospital assays just aren't calibrated for them.
Right, they are too volatile.
But CRP is incredibly stable. It provides a reliable, standardized proxy for systemic central inflammation.
It basically tells you that the IL-6 text messages successfully reached the liver. It gives you a quantifiable number representing the patient's background alarm state.
Spot on. If a patient's high-sensitivity CRP is elevated, the innate immune system is active, DAMPs are likely circulating, and the inflammasomes are firing.
Okay, but this brings us to a mechanical problem. We have a body flooded with CRP, TNF, and IL-1. The systemic alarm is blaring. But we established earlier that the blood-brain barrier, while not an impenetrable moat, is still a formidable physical barrier.
It is.
Cytokines are large, hydrophilic proteins. They cannot just passively diffuse across tight endothelial junctions. So how do they breach the fortress to alter psychiatric function?
It is a multi-pronged invasion. The body has evolved several distinct routes to ensure the brain is kept fully apprised of peripheral inflammation. Route 1 involves the circumventricular organs, or CVOs.
Ah, these are the deliberate architectural gaps in the wall.
Yes. The CVOs are specialized structures clustered primarily around the ventricles. And the capillaries here are fenestrated. They have literal windows.
Wow.
This evolutionary design allows the brain to sample the peripheral blood, monitoring for toxins or endocrine hormones. But it also provides an open door for peripheral cytokines to slip through and bind directly to local neural and glial receptors.
So the cytokines just leak through the windows. But that relies on passive circulation, doesn't it? Which takes time. If the gut is under massive attack, the brain needs to know immediately. That suggests a hardwired route.
And that hardwired route is the vagus nerve. We must remember that the immune system and the nervous system share a fundamental purpose: sensory detection of threats. The vagus nerve wanders throughout the viscera, innervating the gut, the heart, the lungs.
And the sensory fibers of the vagus nerve are physically embedded in the peripheral tissues.
Yes.
And the incredible part, and this always blows my mind, is that these neural fibers are coated in immune receptors. They express toll-like receptors. They express receptors for IL-1.
So when a macrophage in the intestinal lining encounters a stressor and releases a burst of IL-1, that cytokine doesn't actually have to travel through the bloodstream to the brain.
It doesn't.
No, it simply binds to the adjacent vagus nerve terminal. The vagus nerve instantly translates that chemical immune signal into an electrical action potential.
It shoots a telegraph signal straight up into the brainstem, specifically to the nucleus tractus solitarius.
The brainstem receives the electrical warning and immediately initiates a central immune response, perfectly mirroring the peripheral inflammation. All without a single cytokine crossing the barrier.
It's a beautifully elegant neuroimmune bridge. And beyond the CVO windows and the vagal telegraph, we also have the glymphatic system, which has been a major area of recent research.
The glymphatic system serves as the brain's macroscopic waste clearance system. It utilizes a network of perivascular channels formed by astroglial cells.
So it's sort of the brain's plumbing?
Exactly. Peripheral cytokines can enter the cerebrospinal fluid via the choroid plexus, travel through the subarachnoid space, and dive deep into the brain parenchyma through these Virchow-Robin spaces. They diffuse directly into the interstitial fluid, baiting the neurons.
Okay, so the fortress is officially breached, the signals are inside, the alarm is ringing in the central command. Who responds? Who are the local security forces stationed inside the brain?
The primary responders are the microglia.
Ah, the microglia.
You can consider microglia as the brain's resident tissue macrophages, but they are far more sophisticated. I mean, they make up about 10 to 15% of all cells found within the brain.
In their resting state, they are beautifully ramified, aren't they? They have these long, delicate branches constantly extending and retracting, feeling their environment. They act almost like gardeners, tending to the synapses.
They are incredibly attuned to their environment. They possess receptors for almost every known neurotransmitter. So they're constantly sampling the ambient tone of the brain. When neurons fire, there is a natural spillover of neurotransmitters from the synaptic cleft.
Right.
And the microglia monitor this spillover.
So neural activity itself dictates microglial behavior. I've heard them described as bouncers inside a nightclub, where they adjust their level of aggression based on how loud the music is playing and how rowdy the patrons get.
That is a brilliant analogy. The volume of the neurotransmission dictates their phenotype. But when those peripheral alarm signals arrive, whether it's a cytokine leaking through a CVO or an electrical burst from the vagus nerve, the microglia rapidly shift state.
What do?
They pull in their delicate branches. They become amoeboid. They turn into active, aggressive macrophages.
They stop gardening and start fighting.
They begin synthesizing and releasing their own central cytokines. They amplify the peripheral signal. The systemic fire has now sparked a central fire.
And this is the moment. This is where the biology translates into psychiatry, the chemical fallout. We're about to look at how this microglial activation actively dismantles the specific neurotransmitter systems we target every single day in the clinic.
It's quite sobering when you look at the mechanics.
Let's begin with serotonin, the absolute cornerstone of our classical depression models.
The traditional model posits that depression is driven by a deficit of serotonin in the synaptic cleft. And we administer selective serotonin reuptake inhibitors, SSRIs, to block the reuptake pumps,
Forcing the serotonin to linger and enhance signal transmission. It's the foundational logic of primary care psychiatry. But how does inflammation disrupt this?
When the central innate immune system activates, cytokines like TNF and IL-1 trigger an intracellular signaling cascade known as a P38-MAPK pathway.
Mitogen-activated protein kinase.
Yes. And this pathway does something devastating to the serotonin system. It actively increases both the expression and the functional capacity of the serotonin transporter pumps on the presynaptic neuron.
Wait, let me make sure I am framing this correctly for the clinician listening. If I prescribe an SSRI, I am trying to block those pumps. But if the patient is suffering from chronic sterile inflammation, their immune system is using the P38-MAPK pathway to actively manufacture and deploy more pumps.
The immune system is literally building molecular vacuums to aggressively suck serotonin out of the synapse. It is inducing a chemical architecture that directly opposes your pharmacological intervention.
It's like trying to bail water out of a sinking boat with a teacup, while the immune system is using an industrial pump to flood the hull. No wonder the SSRI fails.
The clinical data robustly supports this too. Human neuroimaging studies demonstrate that patients with elevated circulating TNF exhibit increased availability of serotonin transporters in the brainstem.
That's undeniable proof.
Furthermore, if you administer a targeted TNF antagonist like etanercept, which is used in rheumatology, you can actually observe a subsequent decrease in brainstem serotonin transporter availability, which perfectly correlates with an improvement in depressive symptoms.
So we have established a direct causal mechanical link between an immune molecule and the failure of our primary antidepressant class. But the devastation doesn't stop at serotonin, does it? We have to examine dopamine.
Dopamine is the engine of motivation, reward processing, and psychomotor speed. When we look at patients with high inflammation depression, their symptom profile is very distinct. They don't just feel sad.
No, they exhibit profound anhedonia, a total inability to anticipate or experience pleasure.
They show psychomotor retardation. Their physical movements and cognitive processing just slow to a crawl.
To understand why the immune system attacks motivation, we have to go deep into the biochemistry of oxidative stress.
When microglia and macrophages activate, their metabolic demands skyrocket. To destroy perceived pathogens, they generate massive quantities of reactive oxygen and nitrogen species, free radicals.
That's the tear gas.
Yes, this creates immense oxidative stress within the tissue.
And the body has to protect itself from this collateral damage. So it deploys antioxidant cofactors to neutralize the free radicals.
And one of the most critical antioxidant cofactors in the brain is a molecule called tetrahydrobiopterin, or BH4.
But BH4 has a vital day job, doesn't it?
A completely non-negotiable day job.
Yeah.
BH4 is the essential rate-limiting cofactor for the enzyme tyrosine hydroxylase,
which is the enzyme that converts the amino acid tyrosine into LDOPA, the immediate precursor to dopamine.
Without BH4, dopamine synthesis grinds to an absolute halt. BH4 is also required for tryptophan hydroxylase to make serotonin and for the synthesis of norepinephrine.
So we have a brutal physiological conflict of interest here. The body needs BH4 to make the neurotransmitters that allow the patient to feel motivated and engaged with life. But the immune system demands BH4 to survive the oxidative stress of a perceived infection.
It is an evolutionary triage protocol. I often use a wartime economy metaphor for this. Imagine a highly efficient factory that produces luxury sports cars.
Okay, the sports cars representing dopamine and motivation.
Right. Suddenly, the nation goes to war. The government steps in and mandates that all the steel, all the raw materials, must be immediately diverted to manufacture artillery shells to fight the invaders.
The reactive oxygen species.
The factory hasn't forgotten how to build cars, and the machinery isn't broken. But it is entirely starved of raw materials. The consumer economy collapses. In the brain, inflammatory cytokines like interferon gamma and IL-6 ruthlessly commandeer the BH4 supply.
Wow.
They divert it to synthesize nitric oxide and manage oxidative stress.
The body willingly sacrifices the patient's capacity for joy and motivation just to survive what it believes is a lethal threat. The dopamine drought is a direct consequence of this wartime rationing.
We see this played out vividly in iatrogenic models. Before the advent of direct-acting antivirals, we treated hepatitis C with high doses of interferon alpha.
Right, we were artificially inducing a massive viral immune response.
And the psychiatric side effects were legendary. Up to 50% of those patients developed profound, treatment-resistant depression characterized by crushing fatigue and anhedonia.
Because we were stripping them of their BH4.
Exactly. If you put those patients in a PET scanner, you observe significantly decreased dopamine synthesis and metabolism in the basal ganglia. If you analyze their cerebrospinal fluid, you find that the levels of BH4 correlate inversely with the levels of IL-6.
The higher the peripheral inflammation, the lower the central BH4, and the more severe the dopamine deficit. It is a stunning mechanical explanation for the physical paralysis of severe depression.
But we have one more neurotransmitter system to cover, and it is perhaps the most destructive. We have to discuss glutamate and the kynurenine pathway.
Glutamate is the brain's primary excitatory neurotransmitter. It drives neuroplasticity and learning, but it requires exquisite spatial and temporal control. It is a massive double-edged sword.
When glutamate is released into the synaptic cleft and binds to its receptors on the postsynaptic density, it promotes neuronal health, survival, and long-term potentiation.
The good stuff.
But if glutamate escapes the synapse, if it spills over into the extrasynaptic space, it binds to a different set of receptors.
The extrasynaptic NMDA receptors.
Binding there triggers a completely different intracellular cascade, leading to calcium influx, mitochondrial dysfunction, and ultimately excitotoxicity and cell death.
And activated microglia are notorious for disrupting this delicate balance, aren't they?
They are. They release large amounts of extracellular glutamate, often in exchange for cysteine to build their own antioxidants.
But the true villain in this excitotoxic cascade is the kynurenine pathway. This pathway has become the holy grail of neuroimmunology research over the last decade. Let's trace it from the beginning. It starts with tryptophan.
Tryptophan is the dietary amino acid that serves as the raw material for serotonin. Under normal, healthy conditions, a small percentage of tryptophan is converted into serotonin. But when systemic inflammation hits, specifically driven by interferon gamma and TNF, an enzyme called IDO is heavily upregulated in immune cells and glia.
Indolimine 2-cover-3-dioxygenase, IDO. What does IDO do to the tryptophan supply?
IDO acts like an industrial vacuum. It aggressively shunts tryptophan away from the serotonin synthesis pathway and feeds it into the kynurenine pathway. During severe inflammation, over 95% of free tryptophan is hijacked by IDO and converted into a molecule called kynurenine.
Early in the research, scientists thought this was just another way the immune system starved the brain of serotonin, right? The tryptophan depletion hypothesis.
That was the initial assumption. But neurons are remarkably efficient. They can maintain adequate serotonin synthesis even when tryptophan levels plummet. The true danger of IDO activation is not the loss of serotonin. The danger is the kynurenine itself.
Because kynurenine, unlike many other molecules, easily slips across the blood-brain barrier.
It enters the brain parenchyma, and here it faces a critical bifurcation. It can be taken up by either the microglia or the astrocytes, and this creates a devastating chemical tug-of-war.
Let's follow the microglial path first.
In the activated angry microglia, kynurenine is metabolized down a specific enzymatic branch to produce quinolinic acid, or QA.
And QA is highly neurotoxic.
Extremely toxic. QA does three catastrophic things simultaneously. First, it directly stimulates glutamate release. Second, it cripples the reuptake pumps on astrocytes that normally clear glutamate out of the synapse.
Oh, that's bad.
And third, and most importantly, QA acts as a direct, potent agonist at those dangerous extrasynaptic NMDA receptors.
So the microglia are flooding the local environment with glutamate, breaking the vacuum cleaners that are supposed to clean it up, and actively pressing the self-destruct buttons on the surrounding neurons.
It drives profound excitotoxicity, leading to synaptic loss, dendritic atrophy, and neuronal apoptosis, particularly in vulnerable regions like the hippocampus and the prefrontal cortex.
That is the structural brain damage we see in chronic, unremitting psychiatric illness. But you mentioned a tug of war. The brain doesn't just passively accept this toxicity, does it? What is the other path at the K-neuronine fork?
The other path belongs to the astrocytes. Astrocytes attempt to run damage control. They take the kynurenine and metabolize it using a different enzyme to produce kynurenic acid, or KYNA.
Notice the difference in terminology there for the listeners. Microglia make toxic quinolinic acid, QA. Astrocytes make kynurenic acid, KYNA.
KYNA is neuroprotective. It acts as an endogenous antagonist at the NMDA receptors. It binds to the receptors and tries to block the QA and the excess glutamate from causing excitotoxicity.
It is literally throwing its body over the grenade to protect the neuron.
But in states of chronic sterile inflammation, the microglia overwhelm the astrocytes. The balance tips heavily towards QA production. The brain is soaking in an excitotoxic inflammatory soup while simultaneously being starved of dopamine due to BH4 depletion and having its serotonin aggressively vacuumed away by P38-MAPK.
When you lay the mechanics out like that, the idea that major depression is just a mild, vague chemical imbalance seems almost offensively reductive. We're looking at a coordinated, multi-system biological siege.
Which forces us to ask the ultimate teleological question, why?
Okay.
Why would human biology evolve a stress response system that actively destroys our mood, our motivation, and our neuroplasticity? Evolution doesn't tend to preserve mechanisms that make an organism purely dysfunctional.
To answer that, we have to shift from the microscopic to the macroscopic. We have to look at the clinical manifestations through the lens of evolutionary psychiatry, the old friends hypothesis, and the concept of evolutionary mismatch.
We must consider the environment in which our hominid ancestors evolved over hundreds of thousands of years. In the ancestral savanna, what constituted a stressor?
Well, a stressor was acute and almost exclusively physical. You were being stalked by a predator, you were in physical combat with a rival tribe, or you had suffered a severe traumatic wound.
Precisely. A psychological perception of stress, the sight of a lion in the grass, was inextricably linked to an imminent risk of severe physical tissue damage and subsequent bacterial infection.
If you see the lion, you are likely about to be bitten by the lion.
Therefore, evolutionary pressure tightly coupled the psychological fight-or-flight response with the innate immune response. The moment the brain detects a threat and activates the sympathetic nervous system, it sends an immediate hardwired signal to the bone marrow.
It doesn't wait for the bite to happen.
Exactly. It preemptively releases billions of monocytes and macrophages into the bloodstream. It deploys the security forces to the perimeter, the skin, the lungs, the gut, anticipating the breach. It's a brilliant anticipatory defense mechanism.
And if the breach occurs and the organism is wounded and infected, the peripheral cytokines flood the brain. And this triggers a very specific, evolutionarily conserved state known as sickness behavior.
Sickness behavior is a highly coordinated adaptive physiological state. Think about how you feel when you have a severe bout of influenza. You experience profound fatigue, you lose your appetite, your pain threshold drops, you withdraw socially, you experience anhedonia.
You just want to curl up in a dark, quiet cave and be left alone. But why is that adaptive? Why not keep foraging and socializing while you fight off the flu?
Because mounting a massive immune response is the most metabolically expensive action a human body can undertake. Generating a fever requires an astronomical number of calories. Every ADP molecule must be conserved for the immune cells.
I see.
Sickness behavior is a central nervous system program designed to force the organism to stop exploring, stop mating, stop fighting, and dedicate every ounce of available energy to wound healing and pathogen clearance.
It is a forced, life-saving redeployment of metabolic resources. In the ancestral environment, it worked perfectly. You hide, you heal, the inflammation subsides, and the sickness behavior lifts.
But here is the devastating evolutionary mismatch. Our biology has not updated its software to distinguish between a physical threat and a modern psychosocial threat.
Today, my stressor is not a lion. My stressor is a toxic workplace, an impending foreclosure, a contentious divorce, or the relentless pressure of medical training.
The brain perceives the psychosocial threat. It activates the sympathetic nervous system. It signals the bone marrow. It floods the system with monocytes. The cells undergo oxidative stress and leak damps. The inflammasomes fire. The liver pumps out CRP.
The body is preparing to fight a bacterial infection that simply doesn't exist.
And because the stressor, the financial ruin, the toxic boss, never resolves in a matter of days, like a physical wound, the inflammation never subsides. It becomes a state of chronic, smoldering, sterile inflammation.
The central command is receiving a continuous, unyielding signal that the body is severely infected.
Therefore, the brain initiates sickness behavior, and it locks it in. It suppresses dopamine, it alters serotonin, it generates excitotoxic quinolinic acid, the fatigue, the social withdrawal, the anhedonia, they persist for months or years.
Sickness behavior morphs into major depressive disorder.
It is a monumental paradigm shift. We must be clear, though, not all depression is inflammatory. There are non-inflammatory biotypes. But there is a robust, well-defined subtype of depression that is fundamentally a maladaptive, prolonged sickness behavior driven by systemic immune dysregulation.
And clinically, we can identify this subtype. They are the patients presenting with elevated CRP, elevated IL-6, and elevated TNF. Their symptom cluster maps perfectly onto the biological deficits we just discussed: pronounced anhedonia, severe psychomotor slowing, leaden paralysis, and profound sleep architecture disruptions.
And this framework extends far beyond unipolar depression. When we apply this lens to bipolar disorder, the clinical picture becomes deeply sobering. We have to discuss the staging or neuroprogressive model of bipolar illness.
The concept of kindling. The clinical observation that untreated, each manic or depressive episode makes the subsequent episode more easily triggered, more severe, and more refractory to treatment.
We're finally understanding the biological engine driving that kindling. Every acute mood episode generates a massive surge in allostatic load, the cumulative physiological wear and tear of chronic stress.
How does the inflammatory profile of the bipolar patient differ from someone with unipolar depression?
In unipolar depression, the inflammatory markers typically spike during the depressive episode and may normalize during periods of remission. But large-scale meta-analyses in bipolar disorder reveal a far more insidious pattern. Inflammatory markers, particularly CRP and IL-6, are significantly elevated during acute mania. They are elevated during bipolar depression, but crucially, they remain moderately elevated even during euthymia.
Even when the patient appears clinically stable and mood neutral, the biological fire is still smoldering in the background.
The neuroprogression is continuous. The brain is continuously subjected to low-grade oxidative stress, microglial activation, and the slow drip of neurotoxic kynurenine metabolites.
Which explains the progressive cognitive decline, the loss of executive function, and the structural volumetric brain loss we observe in severe, long-term bipolar disorder. The brain is slowly being eroded by its own immune response.
It underscores the absolute necessity of achieving full mucosal and systemic remission, not just a superficial masking of the mood symptoms. But if we want to see the true architect of psychiatric vulnerability, we have to look much earlier in the lifespan. We have to look at neurodevelopment.
Let's shift our focus to schizophrenia and autism spectrum disorder. The immune system is not just a reactive defensive force. It is the foreman overseeing the construction of the fetal brain.
This brings us to a critical concept known as maternal immune activation, or MIA. Epidemiological data stretching back decades has consistently shown a correlation. If a mother suffers a severe infection, particularly during the first or second trimester, the statistical risk of the offspring developing schizophrenia or autism later in life increases significantly.
We are talking about severe systemic infections, influenza epidemics, rubella, toxoplasmosis.
Yes. For a long time, the assumption was that the pathogen itself, the influenza virus, crossed the placenta and directly damaged the developing fetal brain.
But the animal models proved that the virus was entirely incidental, didn't they?
This is the astonishing part. Researchers injected pregnant mice with a synthetic double-stranded RNA molecule called poly-IC. Poly-IC mimics a viral infection. It binds to toll-like receptor 3 and triggers a massive maternal immune response. But there's no actual replicating virus.
It's a purely sterile immune panic.
And the offspring of those mice reliably develop abnormal brain morphology, impaired social behaviors, and sensory gating deficits that perfectly mirror human schizophrenia and autism.
Wow. It is the mother's immune response, not the pathogen, that derails the fetal brain. How does that happen mechanistically?
The maternal cytokine storm, particularly the massive surge in IL-6 and IL-8, crosses the placenta. The fetal brain is incredibly vulnerable to these inflammatory signals. It disrupts neurogenesis, alters the migration of neurons to the cortex, and, most importantly, it fundamentally derails the process of synaptic pruning.
Synaptic pruning is essential. The fetal and infant brain overproduces synaptic connections. To create an efficient, functional neural network, the weak or redundant connections must be aggressively pruned away.
And the cells responsible for pruning those synapses are the microglia. The immune system is the gardener. The microglia use immune signaling molecules, specifically molecules from the classical complement cascade, like complement C4a, to literally tag the weak synapses.
Like painting a target on a building for demolition.
Exactly. The microglia then recognize the C4a tag, engulf the synapse, and digest it. But if the fetal microglia have been primed and thrown into an aggressive hyperreactive state by maternal immune activation,
They don't just prune the weak connections, they run rampant. They overprune. They strip the neural arbor bare.
this aberrant aggressive pruning, driven by hyperactive microglia and upregulated complement proteins, is now considered a core etiologic factor in the development of schizophrenia. The cognitive deficits, the negative symptoms, the eventual psychotic break in late adolescence, they are the delayed, catastrophic echoes of a fetal immune storm.
That reframes schizophrenia entirely. It is a neurodevelopmental immune disorder. But we also see the immune system driving acute psychosis in a much more direct, immediate fashion. We must discuss autoimmune encephalitis.
This represents one of the most triumphant paradigm shifts in modern psychiatry. In these cases, we are not looking at the slow burn of innate inflammation. We are looking at a targeted assassination by the adaptive immune system.
The tactical units go rogue.
For reasons we don't fully understand, sometimes triggered by a hidden teratoma, sometimes following a viral infection, a patient's B cells begin producing highly specific autoantibodies that cross the blood-brain barrier and bind directly to the neurotransmitter receptors on the surface of their own neurons.
The most famous example, brilliantly chronicled in the literature and popular media, being anti-NMDA receptor encephalitis.
A typical presentation involves a young person, often a young woman, who suddenly develops profound acute psychosis. They may exhibit bizarre behavior, severe cognitive dysfunction, autonomic instability, and eventually seizures or catatonia.
Historically, before we understood the mechanism, these patients were misdiagnosed with first-episode acute schizophrenia. They were heavily medicated with antipsychotics, which often made the dysautonomia worse, and they were locked away in psychiatric wards, sometimes deteriorating to death.
They were treated as having a primary psychiatric brain disease. But if a clinician recognizes the subtle neurological signs and orders a lumbar puncture to look for these specific autoantibodies in the cerebrospinal fluid,
The diagnosis flips instantly. It is not schizophrenia, it is a rogue immune attack.
and the treatment is not an antipsychotic. The treatment is aggressive immunosuppression, high-dose steroids, intravenous immunoglobulin or plasma exchange to physically filter the autoantibodies out of the blood.
When you remove the immune attack, the psychosis resolves. The patient, who is catatonic and floridly psychotic, essentially wakes up and returns to their baseline functioning. It is the ultimate proof that the barrier between neurology, immunology, and psychiatry is a complete illusion.
It demands that every clinician maintain a high index of suspicion for autoimmune etiologies when assessing rapid-onset atypical psychosis.
Before we move to treatment integration, I want to ensure we give adequate time to two highly complex manifestations where the immune-brain dialogue is deeply nuanced: trauma and viral sequelae. Let's start with trauma, specifically post-traumatic stress disorder.
PTSD presents a fascinating puzzle. Given everything we have discussed about stress-inducing sterile inflammation, you would expect a patient with severe PTSD to exhibit a massively inflamed profile, both in the body and in the brain.
And peripherally, in the blood. That is exactly what we see.
Yes, large-scale cohorts show that individuals exposed to severe trauma, particularly complex childhood trauma, demonstrate chronically elevated baseline levels of CRP, IL-6, and TNF-alpha. Their systemic alarm is ringing constantly. In fact, some longitudinal studies suggest that high baseline CRP might prospectively predict who is most likely to develop PTSD after a traumatic event, indicating a pre-existing vulnerability.
So the body is on fire. But what happens when we look inside the brain of a patient with PTSD?
This is where the puzzle deepens. When researchers use PE imaging to look at microglial activation in vivo, they use a radioligand that binds to TSPO.
Translocator protein. We should explain TSPO as it's a vital tool in this research.
TSPO is a protein located on the outer mitochondrial membrane. In healthy, resting brain tissue, its expression is relatively low. But when microglia activate and shift into their aggressive inflammatory state, they heavily upregulate TSPO expression. Therefore, high TSPO binding on a PET scan is an excellent proxy for active neuroinflammation.
So in a depressed patient or an Alzheimer's patient, TSPO binding is high. What does the PET scan show in a PTSD patient?
Strikingly, many studies show decreased TSPO binding in critical prefrontal limbic regions.
I want to make sure I am grasping this. The peripheral blood is showing massive systemic inflammation, but the central microglia are showing blunted, decreased activation.
It suggests a profound delinking of the central and peripheral immune responses in severe trauma. The local security forces in the brain might be exhausted or actively suppressed by complex glucocorticoid resistance, while the body's peripheral forces run rampant. It tells us that the microglial phenotype is not a monolith. The inflammation-associated impairment in the synaptic plasticity of the fear extinction circuitry is absolutely present, but the exact mechanism is distinct from classical major depression.
It is a phenomenal area for future research. Now, speaking of profound systemic inflammation, we cannot ignore the greatest global immunological event of our lifetime. The text addresses neurocovid. How does a respiratory virus, SARS-CoV-2, cause such devastating, long-lasting psychiatric and cognitive symptoms?
COVID-19 is a masterclass in how systemic inflammation can wreck brain function without the pathogen ever establishing a massive infection within the neurons themselves.
The virus targets the ACE2 receptors.
Correct. The virus binds to the ACE2 receptors. While there is some evidence of direct viral entry into the brain, the consensus is that the virus primarily infects the sustentacular cells, the supporting cells in the olfactory epithelium.
Which perfectly explains the sudden, profound anosmia, the loss of smell and taste. That was the hallmark of the early variants.
But the severe neurological complications, the acute delirium in the ICU, the microvascular strokes, the encephalopathy are largely the result of the host's exaggerated immune response. The virus triggers an unprecedented cytokine storm.
The systemic fire burns out of control.
This massive surge of cytokines causes severe endothelitis inflammation of the endothelial cells lining the cerebral blood vessels. The blood-brain barrier breaks down. The vasculature becomes hypercoagulable, leading to widespread microvascular hemorrhages and ischemia.
But what about the millions of patients who survived the acute infection, didn't have strokes, but are left with crushing brain fog, persistent depression, and cognitive fatigue months later? Long COVID.
That brings us right back to the mechanisms we discussed earlier. The massive peripheral inflammation activates the IDO enzyme. It hijacks the tryptophan supply. It floods the central nervous system with kynurenine, which the activated microglia convert into neurotoxic quinolinic acid.
The brain is locked into a state of excitotoxicity and BH4 depletion.
The persistent cognitive deficits, the anxiety, the depression, they are the direct legacy of that inflammatory storm, altering the metabolic pathways of the brain.
Okay. We have mapped the mechanisms. We have explored the clinical manifestations. Now, we must transition to integration and clinical reasoning. What does this mountain of data mean for the clinicians sitting in the consultation room? How does this alter the trajectory of psychiatric treatment?
It demands that we embrace precision psychiatry. We must stop treating DSM diagnoses as biologically homogeneous entities. We have to use inflammatory biomarkers to stratify our patients and predict treatment response.
Let's run a clinical scenario. A patient presents with major depressive disorder. You order a baseline high-sensitivity CRP, the lab report comes back, and the CRP is elevated. Let's say it's 4.0 milligrams per liter. What does that single number tell you about your treatment algorithm?
It gives you a powerful predictive tool. Multiple landmark studies, including the GENDEP and COMAT trials, have demonstrated that a high baseline CRP defined as greater than 1 milligram per liter, or greater than 3 in some cohorts, strongly predicts a poor response to standard SSRI monotherapy.
Let me reiterate that because it should change practice today. If your depressed patient has a high CRP, prescribing acetalopram or sertraline is statistically setting them up for fail.
You are fighting the P38 MAPK pathway, which is actively building reuptake pumps faster than your drug can block them. However, that exact same high CRP value predicts a superior response to alternative interventions.
What are the alternatives that work better in the inflamed brain?
Patients with elevated inflammatory markers show significantly better response rates to dopaminergic and noradrenergic agents. Drugs like nortriptyline, or a combination of an SSRI with bupropion.
Which makes perfect biological sense. If the inflammation has depleted BH4 and caused a dopamine drought, you have to directly rescue the dopamine and norepinephrine systems, and SSRI won't touch that deficit. What about interventional psychiatry?
High systemic inflammation, particularly elevated IL-6, strongly predicts a robust, rapid response to both electroconvulsive therapy, or ECT, and ketamine.
Let's focus on ketamine. It has revolutionized the treatment of refractory depression. We traditionally conceptualize ketamine purely as an NMDA receptor antagonist. It blocks the receptor, preventing the glutamate excitotoxicity. But based on our deep dive today, I have to assume ketamine has a direct immunological mechanism as well.
It has a profound, direct anti-inflammatory mechanism. Ketamine does not simply plug the NMDA receptor hole, it actively alters the immune cascade. It rapidly reduces the central production of pro-inflammatory cytokines like IL-6 and TNF.
It puts the fire out.
It goes even deeper. Ketamine administration has been shown to reduce IDO activation, essentially halting the toxic kynurenine pathway at its source. And crucially, it rapidly downregulates the intracellular levels of NF-κB.
Nuclear factor kappa B, the master transcription factor that commands the cell to produce inflammatory proteins.
By suppressing NF-κB, ketamine silences the core genetic engine of the inflammatory response. It is a powerful, rapid-acting central anti-inflammatory agent. This is why it is so uniquely effective in patients whose depression is driven by chronic sterile inflammation.
If systemic inflammation is the culprit, why don't we just borrow from rheumatology? Why don't we prescribe a daily NSAID like ibuprofen or celecoxib to treat depression?
That is the realm of targeted immunotherapies, and the data is a cautionary tale. While some small early trials suggested a slight benefit when adding a COX-2 inhibitor like celecoxib to an antidepressant, the large-scale, rigorous trials have been distinctly underwhelming. Often, there is no significant separation from placebo.
And the risk profile is not benign, is it?
Exactly the opposite. Chronic administration of NSAIDs carries a massive side effect burden. You are looking at significant risks of gastrointestinal bleeding, cardiovascular toxicity, and renal impairment. The minor, inconsistent psychiatric benefit does not justify the severe physical risks. NSAIDs are not a psychiatric silver bullet.
What about the heavy artillery, the monoclonal antibodies that directly sequester specific cytokines, the cytokine antagonists?
The most illuminating data comes from a major trial investigating infliximab, a powerful monoclonal antibody that binds to and neutralizes TNF-alpha. Researchers took a cohort of patients with severe treatment-resistant depression and gave them intravenous infliximab.
And what were the results?
When they looked at the overall cohort, the trial was a failure. Infliximab performed no better than a saline placebo.
So targeting TNF didn't work.
But then they stratified the data. They looked at the patient's baseline inflammatory markers before the infusion. They found a striking bidirectional interaction. If a patient entered the trial with a high baseline CRP, specifically greater than 5 milligrams per liter, indicating severe clinical inflammation aortic cells, infliximab produced a robust, clinically meaningful antidepressant response.
The drug worked beautifully, but only in the inflamed patients.
But here's the critical warning. If a patient entered the trial with a low baseline CRP, meaning they had a non-inflammatory biotype of depression, giving them the powerful anti-inflammatory drug actually made their depressive symptoms slightly worse.
Why would lowering inflammation make depression worse?
Because the brain requires a baseline physiological level of cytokines for normal function. TNF and IL-6 at low physiological levels are essential for long-term potentiation, memory consolidation, and neurogenesis. If a patient is not inflamed and you chemically crush their baseline cytokine levels to zero, you are inducing cognitive dysfunction.
That trial is the ultimate proof of concept for precision psychiatry. You absolutely cannot prescribe a biological immunotherapy without first phenotyping the patient. You must prove the target exists before you fire the arrow.
We must treat the underlying biology, not just the DSM label.
Beyond pharmaceuticals, the textbook highlights 3 massive, modifiable lifestyle factors that dictate the tone of the immune-brain axis. The big 3: obesity, the microbiome, and sleep. A clinician cannot ignore these. Let's start with obesity.
We must discard the idea that adipose tissue is just inert energy storage. Adipocyte fat cells are highly active endocrine and immunological organs. They are essentially factories for inflammation.
How so?
As adipose tissue expands, the sheer physical mass outstrips its local blood supply. The adipocytes become hypoxic, they undergo stress, they die, and they leak damps. This summons millions of macrophages into the fat tissue. A high body mass index drives a relentless, chronic, low-grade sterile inflammation.
It completely blurs the line between metabolic syndrome and psychiatric illness.
If a patient presents with severe depression, a BMI of 38, and an elevated CRP, you cannot easily separate the inflammatory effects of their psychosocial stress from the inflammatory effects of their visceral fat. If you want to lower their central microglial activation, prescribing an antidepressant while ignoring their metabolic health is fighting a losing battle. Weight management and metabolic normalization are primary psychiatric interventions.
The second massive modulator is the gut, the microbiome.
The enteric nervous system is incredibly vast, and it is in constant, intimate contact with the trillions of bacteria residing in the gut lumen. Returning to the old friends hypothesis, we evolved in a symbiotic relationship with these microbes.
They aren't just hitchhikers, they actively manage our immune system.
Healthy, diverse gut flora produce short-chain fatty acids, like butyrate, which induce the formation of regulatory T cells. These regulatory cells provide a constant, low-level, anti-inflammatory tone, keeping the aggressive innate immune system in check. They prevent us from attacking ourselves.
But modern life is a microbiome-destroying machine, isn't it?
The Western diet, heavily processed foods, ubiquitous antibiotic use, and chronic stress decimate the diversity of our old friends. But chronic stress does something even more dangerous mechanically. It induces what is colloquially known as leaky gut.
Explain the mechanism there.
Severe psychological stress activates the sympathetic nervous system, which alters the motility and blood flow of the gut. It degrades the tight junction proteins like zonulin that seal the gaps between the epithelial cells of the intestinal lining. The barrier becomes permeable.
And what leaks through?
Bacterial products, specifically lipopolysaccharides, or LPS, from the cell walls of gram-negative bacteria. LPS slips through the weakened barrier and enters the systemic cortical circulation.
And the innate immune system's toll-like receptors are explicitly designed to hunt for LPS.
The liver and the patrolling macrophages detect the LPS in the blood, and they instantly conclude that the body has suffered a massive, life-threatening bowel perforation. They trigger a systemic alarm, shooting IL-6 and TNF levels through the roof,
which then cross the blood-brain barrier, activate the microglia, steal the BH4, and cause severe depression.
Therefore, restoring the integrity of the gut barrier through dietary interventions, prebiotics, and stress reduction is not just about digestion. It is a targeted psychiatric intervention designed to seal the perimeter and stop the false alarm.
Which brings us to the final and perhaps most immediate modulator, sleep.
Sleep architecture and immune function are inextricably linked in a bidirectional feedback loop. Under healthy conditions, the immune system follows a strict circadian rhythm. Your systemic levels of pro-inflammatory cytokines, like IL-6, naturally peak during the night, particularly during slow-wave sleep.
So the body conducts its immunological maintenance while you are unconscious.
Exactly. Sleep regulates the distribution of circulating lymphocytes and the consolidation of immunological memory. But what happens when sleep is chronically disrupted?
Which describes nearly every patient walking into a psychiatric clinic. Insomnia is ubiquitous.
Sleep deprivation completely, violently inverts the cycle. It blunts the normal, restorative nighttime peak of cytokines and instead causes high, persistent, abnormal inflammation during the day.
So the patient is walking around flooded with IL-6 while trying to function.
And the genomic impact is terrifyingly rapid. Studies have shown that just a single night of partial sleep deprivation induces epigenetic changes in peripheral immune cells that are characteristic of accelerated cellular aging.
Inflammaging.
Yes. Now consider the aging brain. As we age, our microglia naturally become primed and hyperreactive due to the slow accumulation of misfolded proteins like amyloid or tau. If you take an elderly patient with primed microglia and you add the systemic daytime inflammation caused by chronic insomnia,
you throw gasoline on the smoldering fire.
You dramatically accelerate the process of neurodegeneration and cognitive decline. Conversely, in patients with pre-existing autoimmune conditions like rheumatoid arthritis, the sudden onset of a sleep disturbance will almost invariably precipitate an acute depressive episode.
So when a clinician prescribes a targeted sleep intervention, whether it's prescribing melatonin or conducting a rigorous course of cognitive behavioral therapy for insomnia, they are not just trying to make the patient feel more rested, they are actively, mechanically lowering the patient's daytime cytokine load. They are treating the neuroinflammation.
It is all interconnected. You cannot treat the brain without treating the sleep, the gut, and the metabolic system.
This has been a truly profound exploration. We have thoroughly dismantled the fortress. To synthesize our journey, the boundary between physical etiology and psychiatric illness is a biological illusion. We have seen how peripheral cytokines breach the central command and actively alter the structural architecture of our neurotransmitters. We have explored how the body ruthlessly sacrifices dopamine to fight oxidative stress and how it weaponizes glutamate through the toxic kynurenine pathway.
We have fundamentally reframed depression. It is not merely a chemical imbalance of unknown origin. It is, for a distinct subset of patients, a prolonged maladaptive sickness behavior driven by systemic sterile inflammation. We have traced how maternal immune activation disrupts fetal microglial pruning, setting the stage for neurodevelopmental disorders, and how unremitting allostatic load drives the progressive kindling of bipolar disorder.
The ultimate actionable lesson for the clinician is that modern psychiatry must integrate systemic biology. When you assess a patient, you must look at their CRP, their BMI, their sleep architecture, and their microbiome, because their immune system is actively and continuously shaping their mind.
And to reinforce that clinical reasoning, I want to address the learners directly. I'm going to challenge you with 5 specific single best answer questions, drawn directly from the mechanisms we've unpacked today.
Please pause the audio if you need a moment to formulate your answer.
Question 1. What is the specific biochemical mechanism by which chronic immune activation directly causes a reduction in dopamine synthesis, leading to the clinical presentation of anhedonia?
Take a second to think.
The answer is that immune activation causes massive oxidative stress, which rapidly depletes the vital enzyme cofactor BH4. Without BH4, the enzyme tyrosine hydroxylase is unable to synthesize dopamine. Question 2. Within the kynurenine pathway, which specific neurotoxic metabolite, primarily produced by activated microglia, acts as a potent agonist at extrasynaptic NMDA receptors, driving excitotoxicity.
Remember the tug of war.
The answer is quinolinic acid, or QA. Question 3. A 35-year-old depressed patient presents with a baseline high-sensitivity CRP of 4.5 milligrams per liter. Based on current clinical trial evidence, are they statistically more likely to achieve remission with an SSRI monotherapy, such as escitalopram, or with an agent possessing NMDA antagonist properties, such as ketamine?
This one changes practice.
The answer is ketamine. A high baseline CRP strongly predicts non-response to standard SSRIs, but predicts a robust, rapid response to ketamine, which possesses both NMDA blocking and central anti-inflammatory properties. Question 4. From an evolutionary psychiatry perspective, what is the adaptive purpose of sickness behavior, and why does it frequently become a maladaptive pathology in modern society?
The old friends and the lions.
The answer is that sickness behavior evolved to conserve massive amounts of metabolic energy to fight physical pathogens and heal wounds by enforcing rest, isolation, and anhedonia. It becomes maladaptive today because the immune system is triggered by chronic psychosocial stressors, where there's no physical wound to heal, leading to a persistent, unresolved state of behavioral shutdown, clinically recognized as depression. And finally, question 5. Which early developmental event, often triggered by severe maternal viral infections during gestation, is strongly associated with a significantly increased risk of the offspring developing schizophrenia, primarily via the disruption of microglial synaptic pruning?
The final puzzle piece?
The answer is maternal immune activation, or MIA.
Those are phenomenal questions to anchor the clinical reasoning. As we conclude this deep dive, I want to leave you with a final provocative thought to carry into your next clinical encounter. Think about the implications of epigenetic memory. If the physical architecture of a patient's mind is being continuously pruned and shaped by their immune system, and if their macrophages and microglia are holding on to the epigenetic echoes of every viral infection, every period of metabolic stress, and every severe psychological trauma they have ever endured, at what point does immunology simply become the physical study of our psychological baggage? Here are the details of this episode.
Immune-brain interactions in psychiatry. Byline. When defense becomes dialogue, how the immune system shapes mood, mind, and vulnerability.